Here I'd like to talk about a short summary of our poster presentation. The title is Variation in Effect of Host tree Size on the Species Richness and Abundance of Vascular Epiphyte Assemblages Among Tropical Forests. Firstly, the introduction. The vascular epiphytes contribute to local plant diversity in tropical rainforests, and they have high existence functions. Within a forest, the number of epiphyte species on a tree and the number of epiphyte individuals on a tree generally increase as tree size increases. However, a few studies have focused on the relationships in the lowland forest in Southeast Asian tropics, and the variation of these effects has been unclear among the tropics. Therefore, in this study, we aim to clarify the tree size effects on species richness and abundance of vascular epiphytes in bony and lowland tropical forests. Then, we provided a review of among such variation in the regulation relationships between epiphyte richness or abundance and tree size by comparing the results of bony and lowland and those of the other tropical forests. Here, I explain the methodology. Firstly, tree size effects on species richness and abundance of epiphytes were analyzed based on the census of epiphytes on 430 trees in primary lowland forest in Rambil Hills National Park in Borneo. Second, then we compared regulation relationships of our results with those from similar studies done in tropical rainforests. Moving on to the results. Here I only explain a section of this study, the relationships between epiphyte richness and the DBH. The number of epiphyte species increased with DBH increase. The regression line rapidly increased when the DBH reached about 40 cm. Then the line exponentially increased without saturation. Then we compared the results from other sites. The trend found in Borneo was, however, not consistent across other study sites, although the results of a study from Panama, shown as Figure D, had similar trend with Borneo. The regression lines in the results from another Panamanian site, Mexico and China, shown as A, B, C in figures respectively, linearly increased and were different from Borneo. And the results from two mountain sites, shown as B and C, had clear saturation, and this was also different from Borneo. Further, in Borneo, smaller trees with DBH 40 cm or less rarely hosted epiphytes with more than 3 species, and this trend was consistent with the results of a study done in lowland Panama. However, in 3 sites in Neotropics and 3 Southeast Asian tropics, shown as A, B, C in figures, trees with DBH less than 20 cm frequently hosted more than 5 epiphyte species, and it was considerably different from Borneo and lowland. Although the reasons for the differences in this study remain to be determined, examining the relationships of the trends with some factors generally controlling epiphyte distribution, such as, for example, host tree taxa, forest dynamics, climate, and light availability, might help deeper understanding of these differences. The number of studies across the tropics, however, remains insufficient. Furthermore, studies targeting many sympatric tree species in the forest are also extremely limited, and further investigation is needed. In any case, in bony and lowland forests, thicker and taller host trees may importantly contribute to maintaining the diversity of epiphyte assemblages than some forests compared in the study, and such trees are important for epiphyte conservation.